I think just in case was like
Oh,
third.
that's 84 day.
All right, afternoon, everybody. All right. When was that class? Just like yesterday.
Not today though. He
Oh,
was like old camera or it was
okay.
like yeah.
All right. Couple reminders. We're getting here near the end of the semester, but exam number 4 is Friday. Okay.
I should probably start.
And then also the unknowns project is also due Friday. Right. And so we then have our final exam Wednesday, December 10th. And don't forget about the extra credit opportunity for the IDEA course evals.
What's it at now? Isabel said she was gonna do.
I haven't checked it since yesterday, but it was like one person hadn't filled it out as of yesterday.
I have to Isabel to fill it out.
Okay. All right. There is also a Brightspace survey to complete as well, so don't forget that. I did email out that earlier. That should be working. Yeah, okay. I'll email out about that again just to make sure. Okay. Yesterday, we kind of talked a lot about immunity. We talked a lot about virulence factors. And so, for instance, one thing we talked about yesterday was exotoxins. What are exotoxins?
They're
The bacteria makes and secretes an enzyme that targets host cells in some way. And we talked, for instance, some different examples of exotoxins, such as, for instance, hemolysin or AB toxins, for instance. When it comes to AB toxins, which part of an AB toxin is the toxic part, the A subunit or the B subunit part? It's the A subunit that's the toxic part. The B subunit is a delivery vehicle, for instance. Okay. What we're going to talk about today is instead endotoxins. And these are something we introduced a little bit towards the end of the last lecture. Come here. Okay. There we go. But we're going to talk a little bit about endotoxins. Endotoxins, these are not secreted enzymes. They're called an endotoxin because they're inside the bacterial cell and they're part of the structure of the bacterial cell itself. So they are usually a structural component of the bacterial or pathogen itself. Okay, so they only then get released. They're not secreted enzymes. They only get released if the pathogen dies, breaks up into fragments, and then that can release these endotoxins. And an example of an endotoxin is, for instance, lipopolysaccharides, or LPS for short. There are other kinds of endotoxins as well, but I'm going to use lipopolysaccharide as an example of an endotoxin. Where do you find lipopolysaccharides? In the outer membrane. I know, but one of you said outer membrane. Outer membrane of what? Of gram-negative bacteria. So right here is, for instance, my lipopolysaccharides. But lipopolysaccharides are specifically found in the outer membrane of gram-negative bacteria, and that's the only place they're found. Lipopolysaccharides are specific to gram-negative bacteria, and only gram-negative bacteria have these lipopolysaccharides. It's a structural part of then the outer membrane. This part right here is called the O antigen. So this entire thing is a lipopolysaccharide. This O antigen part, this is on the surface of the outer membrane. That is not the toxic part, the endotoxin part of a lipopolysaccharide. That O antigen is on the outer thing, and this is just a pattern of sugars that changes from one species of bacteria to another to another. This part is not the toxic part. It can react with antibodies. It can do some other stuff that we talked about. However, there is a part right here that is called the lipid A portion. And that lipid A portion is this region down through here. But typically, this part is membrane embedded. So it's inside of the outer membrane. And if it's in the outer membrane, it's actually usually not visible to our immune system. Our immune system can only detect surface molecules, things that are presented on the outside. It has trouble seeing inside of cells or inside of membranes. And this is going to be membrane embedded in the internal portions of the outer membrane. So typically our immune system is usually blind to that lipid A portion. However, when the lipid A portion gets revealed, it can be extremely toxic to us. Now, why is that the case? It's actually not anything to do with the lipid A. This part right here is not inherently toxic. It's not like this goes around and damages things. Turns out our immune system can overreact to lipid A, and then that causes damage. It's an overreaction of our immune system that causes the damage. So how does this work? First off, it's really bad to have a bacterial infection of your blood. Like really bad. If you got a bacterial infection of your blood, it's called septicemia. Septicemia is really bad because that can be the bacteria make their way to the bloodstream, but then the bacteria go through your bloodstream to all the parts of your body, and then you get a systemic infection. So bloodborne infections, bad in essence, because they can spread throughout your entire body. So we have a monitoring system for this. And so there's a monitoring system that's specifically going to be monitoring for gram-negative bacteria that gets into our blood, and then that provides a fast response. So say you have a bacteria, and this is a gram-negative bacteria. We know it's gram-negative because it has these lipopolysaccharides as part of its outer membrane. I'm just trying to do this one. What we have is we have a monitoring system for those lipopolysaccharides. And it's actually a system called lipid A binding protein. Okay, so this is a lipid A binding protein is a host cell protein. Okay, and it's called lipid A binding protein because what it binds to is the lipid A portion of lipopolysaccharides. This is secreted by white blood cells in our bloodstream, and there's just always some of this lipid A binding protein circulating around in our blood. What lipid A binding protein does is it binds to the lipid A portion of a lipopolysaccharide. But those lipid A portions of lipopolysaccharide, those are only visible if a gram-negative bacteria dies, breaks into fragments, and those fragments will then be floating around in the bloodstream. So gram-negative bacteria has lipopolysaccharides in it, it dies, fragments of it, including those lipopolysaccharides, get released, and now that lipid A portion becomes visible. When that lipid A portion is visible, it binds to lipid A binding protein. This then is going to bind with a receptor on the surface of certain white blood cells. And here's kind of lipid A, it's lipid A binding protein binds to that receptor. That sends a signal into the cell and it activates that white blood cell. And then what happens is that white blood cell starts making cytokines. What are cytokines? That's one of the things they can do, but what in general is a cytokine? They're involved in capillary dilation, but that's not a good definition of a cytokine. T helper cells do make cytokines, but then what's a cytokine?
They help kill bacteria.
They do not help kill bacteria directly. Signaling proteins. Cytokines are signaling proteins that, no, he wasn't, that control immune responses. Okay, so these are signaling proteins that control immune responses. We've talked about them a few times now. In the case of cytokines, the white blood cell starts making cytokines. Those cytokines then turn on immune responses. That's what they do. They control those immune responses. And what will end up happening is, yes, the cytokines, one of the things they do is they cause inflammation and then dilation of blood vessels. So we go from our normal size one to our dilated blood vessel. Normally, that is a good thing. Inflammation normally allows more white blood cells to enter to an infected area. It's a bad thing when you have a bloodborne infection. Because if you have a bloodborne infection, is there a localized area or is there a systemic area? Systemic, widespread. And if you have a lot of white blood cells that are making cytokines here and here and here and here and here, all throughout your body in different parts of your circulatory system, what happens across your entire circulatory system? All of it dilates, or a good chunk of it dilates. And so what ends up happening is sometimes your immune system is too sensitive to detecting that lipid A, too many cytokines get released, and then the inflammatory response causes all of your blood vessels to dilate simultaneously. And if all your blood vessels suddenly get bigger, what happens to your blood pressure? It drops precipitously. And you go to very low blood pressure, which is then shock, in essence. Shock, one of the hallmark characteristics of shock is very low blood pressure. But without blood pressure, are you going to be getting enough oxygen to your cells? Nope. Are you going to get enough nutrients to your cells? No. The main problem is going to be the oxygen. And so your cells have to start getting trouble getting oxygen. Blood pressure crashes, and you go into something called toxic or septic shock syndrome.
So like when you get like, can you get like high blood pressure from like an infection like that, or it like blocks the production?
You can get high blood pressure as well, and there are some certain chemicals that can do that. That's more of a hormone response and some other things. The other thing, though, is an infection typically ramps up white blood cell production. And so you get more white blood cells circulating around, and the blood pressure typically goes off to deliver those to target areas a little more quickly. With higher blood pressure, the blood's going faster as well. So there's some physiological responses. A lot of times people with an infection will naturally have higher blood pressure, but it's a physiological response. Questions about that? Okay. Septic toxic shock, there's about 20 million cases of this a year worldwide. Symptoms for this are high fever, hypotension, low blood pressure, high heart rate, kidney failure, liver dysfunction, and then changes in mental status. A lot of times people that are going into toxic shock, they start getting confused easily and like aren't like short-term memory issues and things like that as they go into that shock response.
Like last over like a long period of time. That's like a quick.
Well, the problem is if you go into shock, it's bad. I mean, like it has a mortality rate of 20 to 50%. Kind of depends on where you are. If you're not in a hospital already, it's around the 50% mark. If you are in a hospital and then you do some quick intervention, it's 25%. But either case, it's bad. Okay. It can rapidly lead to death. And this is the #1 cause of death in intensive care units, ICUs. There has been increasing rates over the last few decades. Anybody know why? Nope. Antibiotic resistant bacteria. So there are more bacterial infections that don't respond to antibiotic treatment. And then with higher rates of bacterial infections, you get higher rates of toxic shock syndrome. All right, last topic for today is then antibiotics. Okay, antibiotics were first discovered. Anybody know when they were first discovered? Oh, it's on the slide. All right, fine. They were first discovered in 1928 by Alexander Fleming and completely by accident. Alexander Fleming was doing research on Staphylococcus aureus and staph infections. He was growing Staphylococcus aureus on a plate. He was kind of notorious for having dirty labs. Like he just didn't have very good cleaning and tends to just be really cluttered. Well, he had a fungal contaminant land on the plate, and he was just going to toss out the plate, but then he noticed this, that the bacteria was applied there, but near the fungus, was the bacteria growing near the fungus? No, he was actually noticed that the fungus, in this case, the fungus that contaminated it was called Penicillium notatum. It is notable because it makes penicillin antibiotic, and that inhibited the growth of the Staphylococcus aureus. He then went on to do research on that, characterized it, and over the course of about 10 years said, hey, this could be useful, but I don't know how to do it, so he abandoned the project. A couple chemists came along who knew what they were doing, and then they actually figured out how to more mass produce the penicillin antibiotic. And by the early 1940s, it was starting to be used to treat disease. Some microorganisms will synthesize and secrete chemicals that then inhibit the growth of other microorganisms. So I might have this guy making something like an antibiotic, and it is inhibiting growth of this other species of bacteria. Why would they do this? Yep, they are killing their competitors. And so the idea here is that they make these chemicals to kill off their competition, then they are more successful. Okay. And almost every single antibiotic we know of was not designed or invented by humans. We found a microorganism that made this antibiotic. We characterized the antibiotic, and we figured out if we could also use it to, say, kill, say, things like bacterial pathogens. So these are almost all naturally occurring substances out in the environment that we found some type of species making.
They're like one, I guess, like range that like inhibits bacteria more often. Like for the fungus was the one for staph. Is that like a more prevalent one?
There's a lot of bacteria that make antibiotics to kill other bacteria. There's a lot of fungus that make antibiotics to kill bacteria. Both. The idea with antibiotics, though, these are going to be some type of chemical that kills bacteria. And that's typically kind of what the loose definition of an antibiotic is. Now, with antibiotics, they also can kind of be broken down into two different subtypes. There's what is called bacteriostatic antibiotics versus bactericidal antibiotics. Bactericidal antibiotics actually kill the bacteria. By the way, what's it mean to be homicidal? Huh? What's a homicide? A killing, a murder. Okay, cidal is Latin for killing, in essence. There is also, though, sometimes what is called bacteriostatic antibiotics, and it actually suspends the growth while the antibiotic is present. So bacteriostatic antibiotics, they don't actually actively kill the bacterial species, they just stop it from growing. Or in essence, they're in stasis, okay? And it stops their growth. This is then what is known as a Kirby-Bauer assay. This is a plate. And what they did is they took it and they just covered it in bacteria. So you guys remember when you did earlier, you did the spread plates? You apply the bacteria to the plate, you spread plate it evenly over it, you create what's called a lawn of bacteria. Right after they were plated and before the cells grew up, they applied discs with antibiotics on them. Okay, and each one of these discs on this plate is a different kind of antibiotic. But we can figure out from Kirby-Bauer assay, are they sensitive to an antibiotic or are they resistant to the antibiotic? Here, this is the bacteria growing up everywhere, including the bacteria grew up right next to the disc. That means this antibiotic did not kill that particular species. The bacteria grew there. Here, though, we see that there was this area of what's called a zone of inhibition or a clearing. And so the bacteria could grow up to the disc, but not all the way. And so the antibiotic was restricting their growth or killing them. And the bacteria didn't grow up in that area. All right. And it creates what are called these zones of inhibition. And those are typically proportional to the sensitivity. That's basically my fancy way of saying the bigger the zone, the more susceptible. The smaller the zone, the less susceptible when I say it's proportional. Okay. Also note, this cannot distinguish static antibiotics from cidal antibiotics. We don't know if this is actively killing the bacteria or it's just stopping their growth. Okay. For instance, something like penicillin stops growth, but doesn't actively kill the bacteria. What is important about antibiotics is they are what is called selectively toxic. We can use antibiotics to treat and kill disease because they are selectively toxic. And what this means, to be selectively toxic, they have to prevent the growth of some organisms, in this case bacteria, but permit the growth of others, in this case humans. So for instance, say you have an active infection in your body. Could you treat that infection using bleach? If you say had a bacterial infection of your GI tract, could you drink some bleach to kill that bacterial infection?
Good, but then you die.
Yes. Is bleach selectively toxic? No.
No, I thought you meant like getting into other bacteria.
Yeah. Bleach will kill the bacteria, but it will also kill you. And in fact, equally well, if not even better, sometimes. So you can't use bleach, and no, you can't even. You should not even be using bleach on your skin or anything like that. Okay, you definitely should not be drinking or ingesting bleach because you're going to kill yourself. Bleach is not selectively toxic; it's an equal opportunity killer. Okay, but antibiotics are. Antibiotics will kill and target bacteria, but just bacteria, and they will not then target human cells. So this is why we can use antibiotics to treat bacterial disease. But to be selectively toxic, the antibiotic has to target some type of specialized function in a cell. Typically, you have to have some type of antibacterial target. You have to target a system the bacteria requires to grow and survive. But you also then have to target a pathway or a system that the human cells do not use, or that human cells have a similar but substantially different version of. So you need something the bacteria needs, has to have it, but that human cells don't have to have that. There's a simplification there. And also some complex multi-step processes are particularly attractive targets because then you might have multiple different antibiotics targeting that process at a different step. You might have antibiotic targeting step 1, different antibiotic targeting step 2, a different antibiotic targeting step 3, for instance, in a multi-step process then. So that can provide many targets for that. What might be an example then of a good target for an antibiotic? What's something that bacterial cells have and human cells don't? Yeah, really good target is peptidoglycan. Bacteria have cell walls. Do human cells have cell walls? Nope. No cell walls around animal cells. Okay. So that is a substantial target. Do bacteria require their cell walls? Yeah. Okay. So one example of a target is peptidoglycan, and there are several different antibiotics that target peptidoglycan. Penicillin targets peptidoglycan, but specifically it targets the cross-linking step during peptidoglycan synthesis. So with penicillin present, you can't do that little final step in peptidoglycan synthesis where you do the amino acid cross-linkers. Okay. What that means is when penicillin is present, you can't make new peptidoglycan. Vancomycin inhibits sugar polymerization. So we talked about peptidoglycan synthesis earlier in the semester, but at one point the NAM, NAG sugars get added on and you build a polysaccharide sugar. Vancomycin is an antibiotic that actually inhibits it, so you cannot join the new sugar units onto the the polysaccharide chain, and you can't make new peptidoglycan because it inhibits that particular step. Bacitracin is an antibiotic, and this inhibits the bactoprenyl lipid carrier. So remember, you start making the NAGM pentapeptide complex here during this one, attached on the bactoprenyl. At some point, it then flips to the other side of the membrane. We talked about this a few lectures ago. Actually, bacitracin will inhibit that step, so you cannot attach this onto the bactoprenyl lipid carrier. And then you can't make new peptidoglycan. So this is a complex process, has many different steps, but different antibiotics target different steps in the process of peptidoglycan synthesis. Another common target for then antibiotics is translation inhibitors. So this is maybe a messenger RNA being translated by a ribosome. We got my small subunit, my large subunit. There are several different antibiotics like tetracycline, chloramphenicol, erythromycin that actually target then translation in bacteria. Tetracycline blocks tRNAs from coming into the A site of the ribosome. Chloramphenicol actually blocks peptide bond formation so that you can't join amino acids together. Erythromycin binds the ribosomes and prevents them from translocating from one codon to the next codon. So the ribosome gets stuck on the messenger RNA, can't move along it. It can't translate the protein. Questions about that? Anybody know what the problem is with these antibiotics? What do these antibiotics target? That's not the problem I'm going to get at, but they do that too. What are they targeting? They're targeting the ribosomes. They're using antibiotics that are targeting a ribosome. What's the problem with that? We need ribosomes? Yeah, so do both bacteria and humans have ribosomes? Yes. So why are these antibiotics targeting only bacterial cells and not human ribosomes? Huh? They're different. Bacteria have what kind of ribosome? 70S ribosomes. And then what do humans have or eukaryotes have? We have 80S ribosomes. And there are structural differences between those ribosomes. There are some clear structural differences between a 70S ribosome and an 80S ribosome. Turns out these antibiotics we just went through, like tetracycline, chloramphenicol, erythromycin, they only bind to and target 70S ribosomes. They don't target 80S ribosomes. So this is one of those examples where we have a similar but substantially different process. Questions about that? All right. This leads me to an interesting organism for you, Streptomyces coleocolor. This was for a long time mistaken for a eukaryotic fungus. This is it growing on a plate. It has the fuzzy colonies. Usually, if you guys are seeing the fuzzies on a plate, it's a fungus. Not always, though. There are some bacteria that look on like this on the plate, and Streptomyces is an example of that. It's actually, despite the fact that it looks like a fungus, it grows like a fungus, under a microscope, you're going to mistake it for a fungus because it grows in these hyphae patterns, which is usually a fungal growth pattern. But it is just an example of convergent evolution, where it has evolved over time to have a similar type of features as a fungus. Okay. But it is a gram-positive zoobacterium. It's unusual in that it also has a linear chromosome. But most bacteria, do they have linear chromosomes or circular chromosomes? Usually bacteria have a circular chromosome, almost always. This is an example of a bacteria that has a linear chromosome. There's always some exceptions to the rule. It has a complex developmental cycle. It includes some spore production that makes these aerial spores that are going to be dispersed by the wind. Again, that looks like a fungal response, but it's not. Fungi also make these aerially these spores that are dispersed through the air. But this is another example of convergent evolution, not actually relatedness. Then. It also makes antibiotics, and it turns out a lot of antibiotics. Ivermectin, Biolafis, bleomycin, globomycin, kanamycin, chloramphenicol, streptomycin, erythromycin, vancomycin. All of these are antibiotics that are made by some type of Streptomyces species. And about 75% of the world's antibiotics come from some type of Streptomyces species. Okay, so there's a lot of research on this guy. There's a lot of attempts to find new Streptomyces species that might be making new antibiotics. Each different species of Streptomyces makes often multiple antibiotics. Each species often makes its own unique version of antibiotics. For instance, Streptomyces avermatilis makes avermectin. It's actually named after the antibiotic it made. Okay, questions about that? Streptomyces, these are some examples of what are called some antibiotics called ketide antibiotics. There's erythromycin, globomycin, vancomycin. Do they look pretty different compared to one another? Yeah, and these are some complex molecules. Turns out they're all modified fatty acids. And a lot of antibiotics are just modified fatty acid molecules. Complex ones, very complex ones, but they are often related to fatty acid biosynthesis. By the way, what's that mean about often their membrane solubility? Do you think they're membrane soluble or membrane insoluble? They're usually often, not all antibiotics are, but a lot of antibiotics are membrane soluble, where they can pass freely through membranes because they are fatty acid derivatives. Okay. What's going to be actually important here is that there are some genes in the DNA. So this is, say, the DNA. There are going to be some antibiotic synthesis genes in the genome of any type of antibiotic producing species. These antibiotic synthesis genes, they encode enzymes that are going to make and produce an antibiotic molecule. There are typically also going to be some genetic regulators. These just control the production of antibiotic. They turn the antibiotic production on and off. Antibiotic production is kind of expensive. It's not always made. It's made when they detect competitors or during starvation responses, typically. Okay, when they're trying to eliminate their competitors is when they're making it. But here's the also thing. We got a bacteria making an antibiotic to kill bacteria. What's the problem with that? Yep. It can kill itself. What's to kind of stop a bacterial cell from making an antibiotic, and then the antibiotic just comes right back and kills the bacterial cell? That could happen. And that's why typically antibiotic producers also encode somewhere in their genome resistance genes. These resistance genes, again, these are the DNA sequences, they make resistant proteins. And those proteins block the activity of the antibiotic and they protect the cell from that antibiotic. So these antibiotic producers, they're making antibiotics, but they're typically also making some kind of enzyme that protects themselves from their own antibiotic in many cases. Questions about that?
Is there like an antibiotic that like will kill the bacteria that makes it?
Yeah, if you take away the resistance genes and the enzyme that helps protect them, they'll die. If you knock out that gene and they start making antibiotic, they'll kill themselves.
Is it an added gene or do they have it already?
It's evolved. They've evolved to have that already over time.
Are ones that like don't have it, like not very common, to like be an infectious kind of bacteria?
Actually, back in the day, most pathogens didn't have resistance genes. There was no reason for them to have them.
No, I do.
Now, it is fairly common in soil environments, bacterial species that have evolved in soil. It is fairly common for them to have resistance genes. But most of the bacteria we found that make antibiotics or fungi that make antibiotics, it turns out most of them have come from soil environments. There's just a lot of competition for nutrients in the soil. It's not as nutrient rich as a body, so competition is more of a factor. And so there are a lot of bacteria have evolved ways to kill their competitors. And this is where antibiotics come from. Pathogens get into a body, and then it's just like nutrients everywhere. We don't have to worry about competition. Okay, we're just going to eat this giant thing that's us. And they don't have to worry about competition as much once they get into a host. So traditionally, they had not had resistance genes until some things we're going to talk about here in a second. I like weird questions.
If we're allergic to an antibiotic, are we allergic to the bacteria?
No.
or-
Just the antibiotic.
So what makes
You're
it-
allergic to the antibiotic chemical, which is because what has happened is your immune system has actually developed a memory response against the antibiotic itself. It has nothing to do with the bacterial cell.
How is that?
It's in the case of certain antibiotics, kind of odd. For instance, penicillin is a very common allergy. Penicillin is actually one of the rare chemicals that can trigger all 4 types of hypersensitivities. Penicillin can trigger type 1, type 2, type 3, and type 4 hypersensitivities. And it's one of the only things I know of that can trigger every kind of allergy. So, but it's kind of odd. You are giving a high dosage of that particular medicine compound, penicillin. You get a lot of it in your body, and sometimes your immune system just says, what is that? I don't like it, and start developing a memory response against it when you do that.
That's like if you took it like relatively consistently or something.
No, it only takes once sometimes. Yeah, because you're taking a high dose.
That's why you can be allergic the first time and allergic the second time.
You're never allergic the first time.
Yeah.
You're allergic the second time because at some point when you're during the first exposure, you developed a memory response. You're never allergic to anything the first time you get exposed to it. It's the second time. So the first time you touch poison ivy, you're fine. Nothing to worry about. Second time you touch poison ivy, then you might be in trouble. As an example of that. Because that's an allergic reaction as well. That's a type 4 hypersensitivity. Anyways. All right, resistance strategies. How do they become resistant? And we're talking about bacteria being resistant to antibiotics. How do they mediate that particular process then? One is is just one way to be resistant to an antibiotic. Let's just not let the antibiotic into the cell in the first place, what's called exclusion. So for instance, you can just prevent the antibiotic from even entering into the cell. For instance, many gram-negative bacteria, they got their outer membrane. For instance, something like penicillin, this is not membrane soluble. So penicillin hits the outer membrane and it just bounces off. Does it ever reach its target? No, because the target of peptidoglycan is over in here under the outer membrane. The peptidoglycan never reaches the peptidoglycan target. And it turns out penicillin cannot kill gram-negative bacteria. Can it kill gram-positive bacteria? Yeah, just fine. Okay, because their peptidoglycan, they don't have an outer membrane, and their peptidoglycan's on the outside. The other version of exclusion, though, is pumping the antibiotic out, what is called an exporter. And so what happens is antibiotic gets into the cell, like my tetracycline in purple or in pink here, but then there's going to be an exporter. In this case, this exporter is called a tetracycline exporter. And any antibiotic that gets in the cell gets immediately pumped right back out to the environment. So it's an antibiotic pump. Comes in, pumps it out, doesn't kill the cell. Questions about that? Another resistance strategy is inactivation. This is where they can do this in a couple different ways. They can enzymatically destroy the antibiotic. So for instance, I might have a gram-positive bacteria here, but it makes an enzyme called beta-lactamase. Beta-lactamase will bind to penicillin in pink, and then it just chews it up into fragments. But this guy is going to be making this beta-lactamase enzyme. It secretes the beta-lactamase around itself in the environment. And that beta-lactamase enzyme will then actually enzymatically destroy any penicillin molecules it bumps into. And that protects the cell from the penicillin. Another example of inactivation, it's not destroying the enzyme, I'm sorry, not destroying the antibiotic, but modifying chemically the antibiotic. So for instance, there's an enzyme called chloramphenicol acetyltransferase. What do you guys think that chloramphenicol acetyltransferase does?
Transfer something.
Oh, you read them backwards. What's the ASE ending mean? Enzyme that transfers an acetyl group onto chloramphenicol.
Transfer something.
But what it does is it takes chloramphenicol, if it gets in the cell, adds an acetyl group onto it, adds the acetyl group onto it just the right way that it inactivates the chloramphenicol. Chloramphenicol with that acetyl group attached can no longer bind to the ribosomes and no longer kill the bacterial cell. Questions about that? All right. The last antibiotics resistance strategy is just simply be immune, immunity. So for instance, there is certain Streptomyces species that make the antibiotic streptomycin. Streptomycin will bind directly to ribosomes to then stop translation, stop protein synthesis. But it turns out Streptomyces species have mutated their ribosomes in just a such way that the streptomycin can't bind to them anymore. So this is a mutation in the ribosome that makes it so the antibiotic can't bind to it. And this is just an example of what's called immunity. You modify the cellular target of the antibiotic. Okay, and then they're immune to the antibiotic. This last one is super, super rare. But if you have an antibiotic targeting a process, just evolve a completely new way to do that process. This almost never happens, but it has been observed. Like, for instance, a particular way to make a particular vitamin. There's a couple species of, Staphylococcus aureus, and there's an antibiotic called trimethoprim. Trimethoprim. Trimethoprim actually inhibits vitamin synthesis, vitamins that the Staphylococcus aureus needs. Well, there's certain species of Staphylococcus aureus that just evolved a completely new pathway to make the vitamin that's completely different than what it had before, and they're then resistant to the trimethoprim now. By the way, this is super rare because it takes not just one evolutionary event, but a whole combo of evolutionary events to occur. Questions about that? Okay, I hesitate to even bring it up, but it's been observed out in nature. Okay. Resistance genes, the last thing that I want to talk about, how do bacteria become resistant to antibiotics? Again, bacteria make antibiotics that kill other bacteria. Antibiotic producers, though, they have to contain genes that can code resistance to their own antibiotics. But bacteria also like to do horizontal gene transfer. And so what ends up happening is, and especially because resistance genes a lot of times are found on plasmids, but sometimes what you have is you have an antibiotic producer, say the antibiotic producer dies, you got then some of those resistance genes floating around in the environment, but then they get taken up by a pathogen by a horizontal gene transfer. And guess what? Now you have a resistant pathogen. So sometimes these genes hop from one species to another to another. And if a resistance gene hops into a pathogen, you now have an antibiotic-resistant pathogen. And the more we use antibiotics, the more we get resistance then. So this is just a graph of antibiotic usage by country. But you can typically see there's kind of a trend line here. The more a country uses a particular antibiotic, the more you get resistant bacteria. Then as well, pathogens that are resistant to it. Questions about that? And this is appearance of resistance over time for several different families of antibiotics then. There are some species of bacteria, like certain subspecies of Enterococcus faecalis, Mycobacterium tuberculosis, Pseudomonas aeruginosa. There are some species, some subspecies of pathogens that are now untreatable by any known antibiotic that we use to treat disease. So there are some Mycobacterium tuberculosis infections that are untreatable. There was somebody in Wisconsin back in 2010 that had an untreatable version of Mycobacterium tuberculosis TB, and she was put in quarantine until she died so that it did not spread to other people because it was untreatable infection and they didn't want that hopping around in the population.
So like something like that, is it like pretty much guaranteed you're gonna die?
It's not guaranteed. There's always a chance your immune system fights it off, but probably not with tuberculosis. Tuberculosis is slowly progressive, but it has so many defensive barriers around it, your immune system can almost never fully remove it.
Right.
Yeah. And you just get gradually, very, very gradually sicker and sicker. Okay. So last thing for the semester, very last thing for the semester, how do we prevent resistant pathogens? Well, let's just make new antibiotics, except again, we don't make new antibiotics. We discover them. We discover microorganisms making them, and then we co-opt them for our own use. It takes about 20 years to find, develop, and test a new antibiotic. But then once you put it into circulation, it takes about 2 years for the microorganisms to start developing resistance to it. They started using penicillin in 1942, 1943. By 1948 to 1950, they already had the first resistant strains. Way back in the day.
They like mutate and like
Yep.
make so many cells so
Yep.
quickly.
Yep. Bacteria have high rates of evolution, which means they get resistant. Only take bacterial antibiotics for bacterial infections. Do antibiotics do anything for viral infections? Like, can you treat a flu with an antibiotic? No. And remember, antibiotics kill bacteria and only bacteria. Used to be whenever I was a kid, the doctors would give out antibiotics like it was candy. Your parents would take you in, you're sick, here's some antibiotics. Okay. Now they won't give you antibiotics until they do something like a strep test where they do the throat swab and do that one. You have to get the results back before they would prescribe anything. Okay, but you should only be taking antibiotics for bacterial infections. When you're sick, you should take your full prescription of antibiotics. A partial dose will not kill all the bacteria, especially if they're growing in the form of biofilms. Any bacteria that survive a partial dose have a good chance that they might have evolved resistance. Or at least a better chance.
About like the LPS being like defense. Like the LPS being defense, is that like why biofilms are able to survive? Is because they have the LPS?
No, it's just because there's a big group of them. And EPS.
Oh, okay.
Yeah, extracellular polysaccharides.
Yeah, sorry.
Yeah, the antibiotics take a while to get through the EPS layers to the cells because that's a pretty substantial sugar layer around it, and it takes time for them to go down through it. You can take a combination of antibiotics. Well, some bacteria are multi-drug resistant. They may not be resistant to like multiple antibiotics taken at the same time. That might be too strong it might overcome the resistance then. Stop using antibiotics on farm animals. 25 million pounds of antibiotics per year are administered to livestock in the US. Not to treat disease. It's put into their daily feed. It's not a disease preventative. It's not a disease treatment. Instead, for some reason, putting antibiotics into their feed regularly results in almost like a steroid-like action where you get more muscle mass per animal. So you get bigger, bulkier chickens and pigs and cows. And if there's more meat on a cow, the farmer gets more profit per a cow. So but the problem with it is the more antibiotics, also remember that equals more resistance. And so what ends up happening, because you just have a lot of antibiotics on the farm, you tend to then get those antibiotic resistance genes hopping from one species to another to another. Now you got a resistant pathogen. Farmer picks up that pathogen and gets sick, tracks it to the doctor, tracks it to the hospital, and then it starts spreading, it tracks it just into town in general, and then it starts spreading to the populations.
Yes. Are we just in the antibiotics or are they gone? Or-
A little bit. Actually, it's kind of weird. Even when you use the bathroom, there's high levels of antibiotics in your urine. If you're on antibiotic treatment. Turns out also in places like India, they give out antibiotics really regularly in India. There's actually black markets that sell antibiotics in India. They go test the rivers in India for antibiotics and they're everywhere. It's all the people peeing in the river, basically. Anyways. And you get a lot of resistant strains of bacteria come out of India because there's really heavy antibiotic use there that they're trying to get regulated. Last thing is then just put antibiotics on a particular hiatus. If we stop using a particular antibiotic temporarily, we do find that resistance will decrease over time. If the antibiotic is no longer around, what happens over time, the bacterial populations will lose resistance to that antibiotic. So stop taking penicillin for about 15, 20 years. Resistance to penicillin will disappear slowly over time. Questions about that?
You go back to the last slide.
Yes, I can. Let me